everyone for being here. It's after so many years now, like it's it's an important moment. So I wish you can enjoy the presentation. I will present on my thesis of practicing the pot culture, pursuing sustainable agronomic management techniques for the indoor medicinal cannabis cultivation. And see just if it's working. Yes. So we will start the presentation with an outline going through what is actually medicinal cannabis, how it's cultivated in especially indoor systems, the agronomic techniques that we can use through the cultivation that will lead to my 3 publications on the field, followed by a general discussion, and finally an outlook. So first, medicinal cannabis is a sacred plant for many cultures and has coexisted with humankind for millennia. The first references for the medical use for the plant are from the Chinese traditional medicine over 5,000 years ago. And we have seen until today many places in which cannabis cultivation is still traditional, like we see here in Afghanistan or in Jamaica, and more recent in the last decades, some commercial scale production facilities for the supply of these medical markets, as we can see in Canada and South Africa. The plant has spread all over the world due to its multifunctionality, especially across its plant parts. We can cite the use of the stems for fibers, textiles, and paper. We can cite the very nutritional seeds with high contents of proteins and oil. We can also cite, for example, the bioremediation capacity of the roots. But the focus of our research is in the inflorescences and leaves because of the especially phytocannabinoids, special secondary metabolites that are produced in this specialized glandula on the flowers, especially in the female flowers. Cannabis is an annual and short day plant, which means we can play with the photoperiodism to like put the cycles as we wish. So the trichomes are this specialized glandula that produce a bouquet of secondary metabolites, and the reaction to abiotic stresses is still scarce on the literature how these secondary metabolites are produced or synthesized in for the plant's defense and protection. 2 phytocannabinoids that I would like to cite, which is the most known ones, are the THC, which is known for its psychoactive effect, and CBD, which in the last years have been used for therapeutical applications. Cannabis also produces a big range of terpenes and flavonoids. And the indoor cultivation system is the system used for the large-scale operations due to the possibility to cultivate the plants year-round by regulating the photoperiodism. This is important, especially in the temperate regions. Also by this. Okay. And due to the protect environment, it's especially important to avoid contamination and extreme weather events, so guaranteeing sanitary conditions for the plants to be classified as pharmaceutical grade, especially under the norms and guidelines from GACP and GMP. Also very important to cite, the indoor cultivation systems are there for the highest control over the cultivation factors, being the cycle duration and the environmental conditions. However, we have to cite that indoor cultivation systems have a very high energy demand and a carbon and water footprint, also producing air pollution and the use or have a strong use of single-use substrates and disposable plastics. Those energy demand is especially related to the energy to power the lights and the air humidification, ventilation, and air conditioning side of this cultivation rules, also for the fertilizer and water usage. It's estimated that 1 kg of indoor flowers represent up to 5,000 kilowatt-stunde of electricity and a range of 2 to 5 tons of equivalent CO2. They estimate the recreational market in Germany in the future can be compared to a total household electricity consumption of the city of Cologne. So going into the environmental factors of indoor systems. We can look into the environment around the plant, so the air temperature, humidity, and the CO2 concentration, and also the characteristics depending on the light that we use, and also the cultivation system itself. So if it's based on a pot culture, hydroponics, aeroponics, and so on. And we can cite management techniques in which people cultivating can apply to manage to change the canopy, the space of the plants, and also root zone management into changing the type of substrate, container size, and so on. So when I started my PhD, the question was, what is known in the literature about these factors? Which factors have the highest significance for yield? And what are the research gaps? So this should let my pathway through the publications, and we will see this every time we change a new publication. So when we look first to cannabis literature in a literature review from 2022, almost 30,000 references were tracked, and more than half of them are from in the fields of medicine and pharmacology, while the other big part is inside the team of drug and public health, and only 5% of cannabis literature in 2022 was related to agriculture. Most of the knowledge is related on cultivation. Agronomy was found in what we call gray literature, so there is a lack of peer-reviewed publications on these cultivation factors and agronomic management techniques. This is when I started my PhD by the end of 2019, and very few was known on the field. So as a thesis objectives, I propose to investigate the impact of these agronomic management techniques on the yield components for the single genotype, name it Canada, in the indoor conditions. When I talk about yield components, I'm referring to the inflorescences biomass, dry weight in grams per plant, the CBD concentration of those inflorescences, and the resulting CBD yield in milligrams per plant, which is the multiplication of both factors. So looking back to the factors influencing the indoor cultivation, what can be observed was my first question, and what can actually be controlled inside my experimental setup. For me, the dimensions of time and space seem the first ones to be relevant, because since indoor cultivation is resource intensive, when we can save time, we can save these resources. And also operating with the light interception of the plant was my first question. That led to my first publication in aiming to optimize time and space. The paper Impact of Harvest Time and Pruning Technique was published in 2022. And first, the hypothesis. So why harvest time is relevant? As I said about resource intensive, there is an accumulation and transformation of those phytocannabinoids during the flowering. So as said, cannabis has a vegetative stage, and once the flowering starts, the phytocannabinoids will be synthesized in the form of CBGA, which will later be transformed into THCA and CBDA. Since there is a high range between genotypes, for my genotype, so what is the harvest time for Canada, was my question, since it's a chemotype 3, a plant that is rich in CBDA. I started from this range and selected 4 harvesting times to conducting this experiment. And when we look into the biomass accumulation over time, the plants continue to accumulate inflorescences until 11 weeks of flower. We can see that the inflorescences at the end are the biggest organ of the plant, the heaviest organ of the plant. However, it seems to be a maximum CBD synthesis and accumulation in these inflorescences, as we see that the concentration goes up and later goes down, which is a possible dilution effect, which means the flowers continue to grow its biomass, but the content of CBD itself do not change. Although not statistically significant across the 4 harvest times due to the high variation, we can observe this trend. And so when we calculate, Tossin, could you maybe drag this thing down so we can... So as we can see here, when we calculate the total CBD yield of the plant, we will find no statistical significance between 9 and 11 weeks of flowering, coming to 9 weeks of flowering as the optimum harvest time for the genotype Canada. And then moving to the second factor, pruning techniques. So the hypothesis is that cannabis is a monopodial species, which has an apical dominance on the main axis and which will form the main top inflorescences on the top. When we apply pruning, we break this dominance and we redirect these hormones to other shoots, in this case, side shoots, when we apply an apical cut. This is utilized in a canopy management strategy to maximize the light capture by having more inflorescences instead of a single inflorescence on the top of the plant. and to avoid the shading of the bottom flowers. So when I look to the plant architecture first, if I don't touch the plants, it will form this monopodial shape with a high accumulation of inflorescences in the bottom and a single inflorescence in the top. For this experiment, I had two treatments, treatment L or lollipop and treatment T or topping. By the treatment L, we have removed the side shoots, so the plant has concentrated all the biomass on the top, whereas in the treatment topping, there was an apical cut at the 7th node, so the plant, the other 6 side shoots grow it longer, and there was more inflorescences in the top and less inflorescences in the bottom. Also, what was interesting to observe, referring to the inflorescences position, first, there is a high variation of the CBD in the inflorescences depending on the position, and that the top inflorescences have significantly higher CBD than the mid and the low fractions. These two are self-shading of the plant. So finally, a CBD yield of the treatments. We can look that 16% higher CBD was found in the treatment topping in comparison to control, although this is not significant, statistically significant. So as an outlook, as a summary, 9 weeks of flowering is the optimum harvest time. The lollipop treatment would concentrate the energy, the biomass on the top of the inflorescences, while applying an apical cut would shifts the biomass from the low to the top, having also more inflorescences in the top. Top inflorescences have significantly higher CBD concentration than the mid and lower fractions, and so we could found 16% higher. Also, it's important to say that the high variance inside the plant, more than 10%, is non-compliant with the BfArM, which is the German health ministry that has the agency of cannabis and define what is the medical quality norms. So in this case, these flowers would be out of the batch. Going to the second publication. So I look not anymore in canopy, but in the root zone management. What is the preferred fertilizer type for cannabis? Can we find similar results with mineral or organic fertilizer? And what is the optimum fertilizer concentration? Also to question, can this stress increase CBD yields? This leads to my second publication in the topics of nutrition and stress. The publication 2, Cannabis Hunger Games, was published last year, and it goes into the nutrient stress induction during the flowering stage. So the impact of these two types of fertilizer using treated fertilizer dilutions on the yield components. So the hypothesis for this is that the metabolism of phytocannabis is already proved to be highly sensitive, either to mineral and organic nutrition. As we know this bell-shaped curve, there is an optimum range. However, these values for the nutrient recommendations in literature are highly variable and depend on the cultivation system and the fertilizer type. We can cite one publication from Bevan that worked with hydroponics and surface response experiment design, and in which he found this particular range for a fertigation system in hydroponics. The other hypothesis of the abiotic stresses like nutrient deprivation, we can see the trigger and production of secondary metabolite in another plants. However, the studies are not conclusive if those stresses can increase the CBD yield without compromising yields. So that's why I had the second experiment on the nutritional stress during the flowering, having 2 factorial experiments, so the type, organic and mineral, and 3 fertilizer concentrations. The fertilizer 240 can be considered as a control or a well-fertilized treatment, and we have 1/3 less fertilizer and 2/3 less fertilizer. This only during the flowering stage. All the plants were treated similarly, the same during the vegetative. So when we look to fertilizer type, mineral fertilizers output significantly higher yields of inflorescences, biomass, and CBD only at the last harvest. However, there is also a significantly lower CBD concentration in the mineral fertilizer. At the end, this can also be a possible dilution effect since the biomass are heavier, and that the limitations in nutrient uptake, especially for the organic fertilizer in the last 2 weeks, which are the most significant, as you can see, the big jump between the last 2 weeks of harvesting, this leads to lower CBD yield concentration, CBD yield results. Looking to the factor of fertilizer concentration, we see here the three treatments, and of course, the more nutrients led to significantly more inflorescences in this case, as we can see by the letters. However, more nutrients in this case led to a significantly less CBD concentration, which can also be a possible dilution effect. So finally, we have a no significant difference in CBD yield between both treatments, as the less biomass would be compensated by a higher CBD concentration. This led finally to a 95% of the same CBD yield, however, using 1/3 less fertilizer. This happens due to the remobilization of already acquired nutrients in other plant parts, which are translocated into the inflorescences during the flowering. So we can see that this inner plant translocation also leads to a higher nutrient utilization efficiency. And we could also observe this while analyzing the NPK dynamics. So we have the NP and K content on the leaves, and we see first that there is a significantly less content of the stress treatments to the control, this across all NP and K. We can see a curve that the nitrogen accumulates in the leaves and later will be translocated into the flowers. We don't know if this is the case for P and K because we see that it reaches a plateau and stays. But finally, we can see, for example, 5 times more N and P in inflorescences than leaves, which means those organs are the most important and the plant will shift everything into these inflorescences. So as a summary from the second publication, the study shows the potential to reduce fertilizer input while maintaining yield. This nutrient acquisition, especially for the organic fertilizer, during the last 2 weeks of harvest led to the reduced biomass and CBD yield compared to the mineral fertilizer. However, the nutrient use efficiency at these lower rates was increased by this large remobilization and translocation of the nutrients. And finally, that we have similar yields using less fertilizer. So moving still on the root zone management, moving to the factor of water. A question of how do plants react under water stress? Can I expect a similar effect of stress in reducing resources during cultivation while sustaining yields? This leads into my third publication on water and stress. The publication Keep Cannabis Blue was submitted to the journal Industrial Crops and Products, and it's currently under revision. And it's based on the hypothesis that medicinal cannabis already shows potential in water-limited environments, as we see some traditional cultivation regions. Studies on the effect of the drought stress on these cannabinoid concentrations and yield are contradictory. We can see the first published paper on this topic in 2019 by Kaplan. He found that applying a severe strain by the end of the flowering led to an significant increase in the concentration of CBD and also in the flower biomass. And we see a result from last year from Duong that found that applying a moderate stress by the early flowering led to a no effect on the concentration and a significantly penalization of the biomass, having 2 genotypes that behave differently to this drought stress. Continuously, the paper that was published this year found no effect of a moderate stress, either to biomass or concentration. However, a severe stress has significantly reduced the flower weight, dry weight and the concentration. So for this experiment, I picked 2 genotypes with different morphology and growth characteristics, genotype Canada and Terra Italia, and applied 2 levels of drought stress intensity. So we have a moderate stress and a severe stress. And I applied this during 3 different stages of flower development, 3 different phenological states during the flower. The control treatment was always maintained in well-watered conditions, and the moderate stress received during 3 times. So at the 3 stress events, there was a moderate stress for this treatment, for these plants, and the severe stress would happen either at the early or at the mid or at the late stage. For the measurements, we measure the pot weight during the water stress, especially in a shorter time period, but also during the whole cultivation, that we can guarantee plants are in a container capacity. So we can calculate the container capacity based on the weight and guarantee that plants are maintained at these levels as a water status level. We also characterized water deficit on the major leaf, which is the largest fan leaf, largest fully developed fan leaf at the end of the vegetative period. by measuring the relative water content and the osmotic potential, also measuring the photosynthetic rate and stomata conductance at this major leaf, and again, the yield components based on biomass and concentration. So the container capacity is this percentage of the substrate water saturation capacity. So when we talk about 100%, we're talking about complete saturated substrate and 0% the completely dry substrate. So for the control conditions, we kept this water container capacity between 60 and 80%. And on a moderate stress, you can see on the red line plants. So this is an example of a water stress event in which at the 0 time plants don't receive water anymore. They are all in a control conditions and will go over time. The plants on moderate treatment would receive a water replenishment not to wilt, and the severe stress plants would wilt completely, not receiving a water replenish. So when we look at the leaf level, less water in the substrate is also translated in less water into leaves, characterized by a lower relative water content of the major leaf. We can see a significantly lower relative water content for the severe stress treatments in comparison to control and moderate, and a no significant difference between moderate and control, which means the plants have a similar water status. This trend is also the same for the osmotic potential, as you can see in the thesis. We have also the measurements of photosynthesis and stomata conductance. So reducing the water status in the major leaf also reduces the photosynthetic rate and the stomata conductance. We can see in the graph the container capacity reducing together with the photosynthetic rate and the stomata conductance. This leads to the metabolic limitations for CO2 uptake and photosynthesis, and we could expect an effect on biomass. So what we see is that the biomass accumulates over time as the results from the first paper. But there is, so until the end of the experiment, but there is no effect of the drought treatments into biomass for the genotype Terra Italia. And we have a significant effect for the genotype Canada, especially lower results for the control and the severe meat, which leads to inconclusive conclusions into this effect. Moving forward, we see no significant effect of the drought stress into the CBD concentration for both genotypes. What we see is a maximum accumulation followed by a dilution effect, as we could observe in the first publication. And as a summary, and this results also translates to the CBD yield. So what we can see as a summary is that our results strengthen the hypothesis that the drought stress does not increase yields, as was proved by Duong and Morgan. And there was a no beneficial effect of applying a severe stress during the flowering stage, as we can perceive no gain in biomass or CBD concentration, but a moderate stress can increase the water use efficiency of plants and reduce the water usage without penalizing yields. So it can be optimized treatments to save resources. As a discussion point, the fang leaves as the major leaf might not be representative for the drought stress measurements at the flowering, as we could observe this water being shifted into the inflorescences. So we arrived to the general discussion. First, it's important to cite that indoor medicinal cannabis indoor systems are currently the standard cultivation system to guarantee safe medicine for people. However, this system, as said, is a resource-intensive system with environmental implications. So further investigations is necessary to reduce the water and energy and carbon footprints of the cultivation with possible pathways. We can look into agronomic management techniques as presented in the 3 publications to increase the efficiencies and reduce the resources, or moving to new technologies or new cultivation systems. So when we look into the agronomy management techniques, this is a summarized table of my 3 publications. We can see that when we look into the factor harvest time and having the treatment of 9 weeks of flowering, we can have a slight, not significant yield penalization when we look in CBD yield per area. However, we are saving 2 weeks of flowering time, so we can save energy and other resources input, like water, fertilizer, and everything that is necessary during cultivation. And when we look into the factor pruning with the treatment topping, we found a 16% increase in CBD yield with an improved canopy management per area, with also a lower variation of CBD because we will have more plants into the top and less into the bottom. Going into the second publication, into fertilization, we can see that the treatment 160, so it's the treatment with a less stress, or with a low stress, 1/3 less fertilizer than control, also led to non-significant yield penalization, however, saving 1/3 less fertilizers. Also, by remobilizing the nutrients, plants can take up nutrients that are in the substrate or in the stems and leaves and will not be used as a harvested material. Finally, for the irrigation paper, we see that the moderate stress led also to non-significant yield penalization. However, this can lead to save some water during the cultivation. And when we look into innovative indoor systems from the industry, we see that some companies are moving into full automation systems, as we can have here in the thesis. I have the chapters, the examples of a cultivation container and also a hottery garden. Those systems have closed water nutrients cycles, so they use the fertilizers until they are done, and have the optimization and verticalization of this space. Also, we can have a citation here of another trend, which is adopting living soil and raised beds. So instead of single-use substrates, they're trying to keep the nutrients and water cycles closed. Or we move into soil sun grow cannabis. 4 other and most GACP active pharmaceutical ingredients of medicinal plants are currently produced in the fields. We can give the example of a rosemary essential oil or opium plants used for the opioids industry. They come from fields. So when we look at the plants growing in the fields in the experiments that we had here last year, the same genotype would have a double yield of flowers and 50% higher CBD in an open field, and would have up to 5 times higher biomass than my indoor experiments when they are grown into a tunnel that also have fertilization and higher temperatures due to the tunnel. We found also double the concentration of CBD and up to 2.6 times higher CBD yield per square meter. Besides this, it's a carbon positive cultivation system, and it's easy to scale up. So, finally arriving to our outlook of my thesis, we need to discuss the decarbonization and sustainable perspective of cannabis cultivation in Germany, since we are at this crossroad in which the law was passed this year, and we still have no ideas how this cannabis landscape will develop in Germany. But we need to look at a sustainable perspective in the 3 pillars of sustainability as a social, economical, and environmental pillars. The indoor cultivation is very important and should be used as a standard only for the medical markets, because people would need this. And the agronomic management techniques can be applied to enhance the sustainability of these indoor systems, as represented in the 3 publications. Additionally, or however, the high variation of the CBD in the flowers found in my experience is unsuitable to be used as a standardized medical products in the way they are used nowadays. So this non-compliant material is also currently being destroyed. So imagine something that costs a lot of carbon and needs to be destroyed afterwards. Medical patients should rely then on standardized extracts that can be produced with the entire plant and plants that come from outdoor cultivation. So we can foresee a German domestic cultivation based on outdoor when it's possible, as we can see for the all other agricultural products, and to receive imported goods from other areas. And when we talk about imported goods, I would like to cite a sustainable future for cannabis farmers. As we can see, can we foresee a strong value chains of imports from traditional regions that have the good environment for the cultivation throughout the year, like cocoa, coffee, other spices? And can we pursue alternative development or a future fair trade cannabis? The German cannabis landscape has changed a lot in the last 5 years since I started my PhD. Sustainability should be at the heart of the current discourses, in my opinion, since the system is changing a lot now, and a lot will be possible. And cannabis could be a solution and not another problem for the decarbonization goals in the coming future. So my list of references, my list of image sources. And I would like to thank you for your attention and for being here today.